It's my pleasure to introduce Carlos Ignacio today, and he's going to be speaking about Mate and Check, which is a practical approach to identifying F1s. And Carlos is an assistant scientist at IRRI who specializes in molecular breeding and bioinformatics. And he's also doing a master's degree in genetics and computer science. Carlos is our Gobi mid-breeder, and for those that don't know that position, it's a position that works with breeders to help with Gobi system adoption and to help develop and train and have adopted the marker tools that are associated with Gobi. And he's been really instrumental in designing our flapjack tools, as well as connecting B4R, which is the breeding management system at IRRI, with Gobi via various sample tracking applications. He also has a lot of experience in designing purity SNPs and F1 pedigree verification panels, and he's designed the purity SNP panel for rice, which is now used across IRRI, also at the Biri Bangladeshi Rice Research Institute, Africa Rice, as well as several rice seed companies. So he's eminently qualified to talk today about identifying F1s, and I will let him go ahead and take the floor. Thank you, Carlos.
Thanks, Liz. Thanks, Star. So today it's going to be just trying to identify F1 hybrids. So this is part of a collaboration with JHI, and we want to interpret low-density markers data for breeders to be able to make decision whether they would need to cull or proceed with a particular plant. Just to make sure that they're proceeding with the right material. So we're first going to define the problem involved and the approach that we are about to take to address the problem, and some benchmarking on the algorithm, both on simulated data and operational data. So I'm going to go ahead and define the current problem. So this is a bit technical definition, but basically out of a genotyping data, let's say from an intertech data, comprised with a very small number of markers and a given set of populations. So among the different populations, which among their samples can be considered as true F1. Problem, there can be many factors that affect on trying to get to a solution. So like what's the rate of genotyping error? What's the rate of missing genotypes, amount of polymorphic markers, the heterozygosity in the parents, basically if they're fixed or not, and if there are, what's the rate that there are off-types, expected off-types in the samples. Also, our current analysis pipeline requires parental genotypes to be included in the dataset. So in this approach, actually we're not gonna use this last one because a lot of our experiences at IRRI that the parents doesn't match the actual population prevents us from actually doing the analysis. So what we would like to do is actually to separate the problem of similarity to parents in a different analysis and just focus on the problem of whether it's really a self or an outcross. So this problem can then be extended later on to a more quality control of how similar the true F1s are to the parents, which is not the scope of this current presentation. So I would just go ahead and try to discuss about how the algorithm works. So I define this as a very naive and greedy algorithm, mainly because we don't know the genotypes of the parents, and we're just basing it plainly on the current data that we have, which is basically the genotype of the population of candidate F1s. And the approach involves some backtracking. So just to give a background, greedy algorithm is a very simple algorithm that you try to capitalize on a uh, certain parameter. So I would discuss that later on what that would be. And backtracking is trying to keep some records in memory in order for you to go back and test whether there are better solutions available to solve the problem. Uh, Next is we're trying to determine the best solution. So this is the greedy approach now from a marker that has a very high number of heterozygous. So we'll go into that later. And additional feature of this approach is that we can check whether a given solution is correct or not. So this is part of trying to figure out the how confident are we with our solution. And I later on I figured out that this is not currently working as expected right now, but there can be a lot of improvements to it. So the limitations of this approach is that it highly depends on the presence of a good marker, meaning the marker should be polymorphic for both parents and they're not segregating for any of the parents. So meaning the parent should be fixed for this marker and that it can be called quite with high confidence. So if there is no polymorphic marker in the data set, then there is pretty much nothing we can do with it when it comes to F1 confirmation. So we cannot do anything without really a good set of markers. And as I mentioned earlier, there should be a good prediction for the segregation pattern, or just to try to predict the expected F1 genotypes. But it greatly depends on how accurate the prediction is. So I'm going to demonstrate step by step how the algorithm works. So as I mentioned, we go for a marker that have very high head count. So those are usually the good markers that would help us identify the true F1s. So in this particular data set, marker 2 and marker 4 have 6 heterozygous skulls. And from those markers, we can arrive to a particular solution. So the algorithm works that you start only with one marker first and try to draw a solution for that. So when we try to consider that this marker is actually a good marker to infer a solution in, then we can just directly translate a heterozygous skull to a true F1. And if there's a homozygous skull, it would directly translate to a selfing line. So it's very simple, and this is pretty much how I do most of my scoring manually. So after this step, actually, we have already a solution, and we can go further onto a next step, which is trying to confirm whether it's a good solution. And if we find out that this is not a good solution, then we proceed with the next marker and so on until we exhaust all of the possible solution offered by this particular data set. So we'll go ahead and test this current solution that we have on the left, which is all of the samples are true F1 except the second and sixth one. So these are all the expected F1s based on the solution, and what we wanted to do is try to guess the segregation pattern of this, meaning that based on their segregation pattern of each marker, we can classify them in different categories, and based on that, we can predict the genotypes of the parents and subsequently the expected progenies. So for this particular example, here is the breakdown of the observed genotypes, and we use a Fisher exact test to determine in which category they fall in, with the expected values here on the left and the Fisher exact test values here on the right. This is, I'm not sure if this is the best test to actually use, but this is better than chi-square because as the number of samples decrease, chi-square is not that accurate anymore, and Fisher exact test does that job more accurately. But I'm in a stage that I'm considering other tests for this particular step. But later on, we can see whether this test is actually good enough for the meantime. So for this particular data set, we ended up with these cases for this marker. So we can then translate this to an expected genotype. So it was only obtained for the maximum values of each marker in which case they match to. So what we're actually looking for is hoping that there are case 3 markers wherein we expected that the markers are polymorphic for both parents and are homozygous. So in this case, there are 3 markers, which is usually a good sign that we'll be able to come up with a good solution here. So proceeding with the next step is trying to figure out the expected genotypes from this different segregation patterns. So it's quite straightforward actually. After knowing what the possible genotypes of the parents are, we would know about the expected genotypes of the F1s. So just to repeat that this is only part of trying to check whether the solution is probably correct solution or not. After this step, there is another step that we can add to that would try to narrow down the possible genotype. So as you can see now, for the expected selfing genotype at the bottom, there are a lot of possible genotypes for these given markers. But if we actually start to make use of the data from the selfing lines, we can actually cancel out some of the genotype possibilities and make this test more accurate. But that approach highly depends whether there are actually selfing lines in the dataset, and will probably fail in cases that there are no selfing lines in the dataset. So we'll just proceed with the first approach now. I'm just showing here now that how to arrive on a more accurate probabilities of genotype of the progenies. So I'll skip that for now, and then move on to the next, where we actually test the observed genotype for the solution we have to the expected one. So we test each sample now, whether they match the expected F1 genotype or self genotype. So we're now in sample 2, testing whether it matches. And then after all are checked, so for this particular genotype data set, we have match already on the first solution. So therefore, if all genotypes of the progenies match, we can accept the solution. Or if there's a certain threshold that the user can input, for example, then that's the time that we can accept this as a solution. But if some of the genotype doesn't match, we can go back to the first step and try another marker for another set of solution until we find a better solution. So basically, that's how we test for the solution. So that explains the algorithm. And the next part is I tried to run simulation test whether how accurate the algorithm is, and based on testing, the runtime actually of the algorithm is highly dependent on the number of samples and number of markers. So the higher the number of markers that we have, the less the more time we actually need to run it. But a normal data set that we actually analyze in Erie is just about maybe 100 crosses and not 1,000. We tested here 1,000 and it took 2 minutes. So I would expect that it would only take less than 20 seconds if we actually run this with an actual data set. So this is also was written in R, and it's running sequentially, meaning one data set after another, but it should be faster if we can compute it at all at the same time or in parallel, and especially if it's written in Java because it can parallelize natively. Also, the other parameters that I've tested was the accuracy of the algorithm. So it's the rate of identifying true F1s correctly. So if it was able to identify for all the samples that we expect them to be to have the same prediction of the true F1s as the simulated one, then it should be considered correct. And then the total number of correct solutions over the total number of simulations, that's what I considered as the accuracy. So the confidence level is, it should tell us about the ability to tell whether the solution is correct or not. So it's computed by the number of percentage of the genotypes of the observed progenies to the expected ones. So actually, I found out that for both of these parameters, accuracy and confidence, I'm not too confident about the results for the confidence, mainly I think because of the Fisher exact test, where it can classify poorly when there's a very few number of samples. So I would just try to focus now more on the accuracy of the algorithm. So here are the different factors that affects the solution. So I've tested 5 of them. I haven't tested yet for missing genotype, errant genotype, and off-type samples because first of all, they are not that common in terms of. I mean, these are the types of factors that we probably don't have control over, and they don't actually have a lot of variance in particular genotyping platform and so on. But we plan to include this test also in the future. But for now, we've looked at the rate of polymorphism, heterozygosity, and number of markers and number of samples, and also the expected purity of the lines, or how fixed the lines of the parents are. So what we observe actually is that among these all tested values, I tried to change across the parameters and held this default values for the other parameters. So If there is high polymorphism, we expect higher accuracy and higher confidence, and that's what we actually observed. And the inverse for higher heterozygosity in the parents led to a lower accuracy and lower confidence. So the for the others, higher of these other three parameters resulted to higher accuracy, but I'm not sure why for the confidence the number of marker affected it negatively. So I really think that the parameter in trying to uh, do the checking of solution should be changed. So maybe try to find another model aside from Fisher exact, or try to make our own statistical model on how to predict segregation more accurately. So I'll try to proceed on to this table wherein it shows the number of correct solutions per a thousand simulation. So the default values are as follows in the white cells, and the yellow ones are the adjusted values. So what we see for the true F1 rate, meaning if the efficiency of a crossing block we expected 90% are true F1s, then we end up of a higher accuracy. And what we actually have in Erie is 90. So we expect that 9 out of 10 of the crosses are true F1s. And if we have that higher crossing efficiency, the algorithm should be much accurate. So the next parameter is the heterozygosity rate. So I've used 30 here, which is I kind of regret using a very high heterozygosity rate, because what we expect our parents to be in is actually around 5 to 10% only heterozygous. But it shows in this test that once we go lower heterozygosity rate, the algorithm actually performs a lot better. So I would expect that if I try to change this het rate for all of these scenarios, that the general accuracy would go even higher than what we see here. So with regards to polymorphism rate, the actual average polymorphism that we have in our purity SNP is about 40%, which is 4 out of 10 markers. And we see that it quickly decays with the number of polymorphism, because this is a very low number of markers, maybe, but it's really important to have a good polymorphism between the parents, and we need to run the correct SNP panel for that. And what we observed with the number of samples, it's quite good actually, that we can drop down up to just 4 samples, but we still maintain a lot of accuracy here. There were some concerns with CIMMYT that they only genotype a few number of lines, so I think it's a really good thing that the accuracy doesn't drop down that much if we decrease from 8 to 4. But at 2 samples per cross, then it completely dies down. And I mean, the number of possible solutions for 2 samples is like only 4 possible solutions. So I would expect a fourth of this. So around 250 is just by chance. So it's not really a good parameter. I would not recommend using only 2 samples for F1 verification and using this approach. So lastly, the number of markers. We have two purity SNP sets that we run. Uh, there's the 10 and there's the 24 SNP, and we can see that once we get as high as 24 SNP, the accuracy really goes higher. So, but the 10 is already good enough, and once we just drop down the hit rate, I'm sure that it will reach 90% accuracy already. So that's it for the simulated data. So I'm not going to talk about the confidence. As I mentioned, it's not really good. I mean, there's no really direct relation between a correct solution and a very high confident, meaning a very high percentage match to the expected genotype. So I'll try to work on that soon. And if anyone can help me in trying to figure out a good model for the prediction of segregation, then I'll be happy to work with you on that. So I'll proceed now to the performance compared to a real data set. So this data set was taken in 2018 dry season in Erie, and in that season, the rice breeding platform has 142 F1 crosses, and per cross we've taken 8 samples from 8 different plants. So the total number of samples, F1 samples, is about 1,100. So what we found out is that so that data set was curated manually by one of the researchers at Erie, and they were able to identify 400 around 460 true F1s. While within that 400, almost all of them were identified too by the algorithm. In addition, there are also other samples that were identified to be true, 441. And for the selfing ones, they were able to identify 224 for both of the algorithms that are selfing. And there's only 13 samples that the researcher considered to be true F1s, but when I checked these 13 lines, it only belonged to 3 crosses, and 2 of those crosses, the algorithm did a better job on classifying those as a selfing lines. So, and the other cross is like only 3 samples. So 3 samples out of 1,100. I think that's a really good accuracy on trying to select which sample should be considered as selfing. So it may not have identified very closely the same number of true F1s, but it did a good job on trying to identify the selfing ones. And trying to see the rate. So this is now the computed efficiency of the crossing. So out of 1,100, there's almost 900 true F1s identified by the algorithm, which is about 80% crossing efficiency, compared to almost half for the curated data set by the researcher. So what we actually observed in a previous study in Erie, the actual crossing efficiency of the crossing block at Erie is around 90%. So the algorithm is much closer to that number, and the 10% difference can be attributed because in 142 of these crosses, around 9 of them, I think 9 to 12 of them, I think 12 didn't had polymorphic markers between them. So they were already automatically scored as self. And those those 12 crosses is about 100 samples, and that accounts for almost 10% for the total number of samples. So I think that's where the difference between the actual observed crossing rate and what the algorithm called differs, and it completely accounts for that. So meaning I think this is this approach is basically fairly accurate and a quick way to be able to say whether a particular sample is an F1 or not, and it can do that without any prior information about the genotypes of the parents. And then secondly, there is a need to improve the confidence measure for the calls, but I think that can be achievable. Just we need to find a good way to predict the segregation. And then there are other approaches that can be tied up with this. A lot of approaches like machine learning can be used to predict through F1s in a quick way as well, because it would only take us a single click for that. And then I would like to encourage the other institutes to work with me and try this on their crops. So it might be a different case for a lot of crops, but I'm willing to work with them to customize the algorithm for them. And hopefully it would work as accurate as what we find now. So I think that ends my presentation. Should I use more time, Star? Liz?
That's fine. You have more slides you could present, or?
Oh, I have 30 slides remaining, but
okay.
I think these are more for details, and I don't think
okay. Let's see what questions you get. Maybe you can go into a couple of the slides that relate to the questions that we have. Does anyone have questions on the line?
Hi, this is Kate. Hi, Carlos. Sorry, I have a little bit of a cold, so my voice sounds strange. I feel very naive, and I think I was also confused by this last time I saw a presentation of this. When you're going through the markers and in your sort of initial table, you have markers with genotype calls of like, or proposed parents, I think, of like AA:AT and like marker 5, 6, 7 or something like that. And I wasn't sure, is that an indel, or that's a single nucleotide, or what's? I didn't understand those examples. I'm sorry.
Yeah, I actually didn't explain it quite well as well. So those are actually the genotype probabilities. So the colon is like just saying that it can the genotype of this particular marker for this parent, for any of the parents, can be either homozygous C or heterozygous. So that's just like possibilities.
Okay, but you put in the case of like the marker 1 just AAAA because it doesn't seem like it could be anything else. Okay.
Yeah, that's correct.
But so in the case of marker 5, why couldn't one of the parents be TT? Oh, I guess. Okay, sorry. If it was if it's selfing, if the AAs represent selfing, then the ATs, couldn't it be AT? Does that make sense?
So because we're assuming here, so the current solution is that they're all through F1 here at the side.
So Carlos, if you go back go back to a few slides more, maybe you can show us the selfing for marker 5.
Yeah, sorry. Okay.
Yeah, go to go to the zeros with 0 and 1. Yeah, from there. Yeah.
So here, yes, marker 5 actually has a TT, but it's for a potentially selfing line.
Mm-hmm. Okay, so that could have been the AT parent that was selfing.
Yep.
Okay. Thank you. Sorry.
Yeah, so actually I have a slide for that tries to explain that we can actually make use of the selfing lines and try to arrive more on a more accurate probabilities whether any of these genotypes belong to which parents do they belong to. So, but I think it's pretty straightforward if someone looks into the slide. But I'm happy to talk to anyone who has questions there through another session or meeting. Anyways.
Hi, Carlos. This is Star.
Yes, sir.
Because this is my second time to hear this, just let me help to translate a little bit the values of your research. It may not be correct, but let me see. I can try it. So I think for a lot of plant breeders, the value of your proposal, also your solutions like this. So you first really have many different crosses, and within each cross, maybe you only have a few F1s or pollinations. And then the workflow, what you are trying to do is, without having the parental lines in the crosses included in your genotyping, you blind folding all the crosses. For example, you have 140 some crosses, right? You look at each cross, if you have 8 samples, within these called sister lines, and you're trying to infer back what are the parents. Then maybe the first step you are trying to see what are the selfings, right? Based on the 0 and the 1s, then you will identify the selfing and the 1s. And the second step, you're trying to infer what could be the parental lines genotypes. That's the second step, right?
Yes.
Yeah. And then the third step, you will see among all the potential F1s, those you call the ones, then you start looking at the markers. So each marker you call that a solutions, is that right? If you have 10 markers, you start looking at the polymorphic markers one at a time, which one will give you the best you call solution. So that part, I'm not fully understand the solution part, but that's my understanding your workflow. You're looking at the markers to see how these markers help you see the segregation fitting the chi-square, but because chi-square limit the independence of the samples, independence of the markers, and also the number of individuals cannot be less than I forgot, is it 25 or 5? In order to meet that requirements, you use the Fisher's test that allow you to count smaller samples. So this is your workflow, right?
Yes, sir. So was there a question there?
I think from like plant breeder try to see the values. I'm trying to make a comment. The reason you're doing this as a Scripps method, the value is you compared at very last second slide. If with a researcher, the curated data versus your algorithm, in addition to the accuracy, the confidence, I'm wondering time to turn around your results, comparing with research, how long you take. Do you have that data?
Yeah, so for this one, actually, I analyzed this. I got the results in maybe less than 20 seconds. So I can actually run this one right now here. So this script, I think, does that. So what this actually does is it reads a Flapjack input file, and that's the one that has 142 datasets. And let's try to clear all the objects. So it would read those files together with the sample file, and if we want to run it, so this is a single run for each population. So that error is because there's a dataset without a polymorphic marker, and I think this will run in just about 20 or 30 seconds. I'm not sure exactly, but that's how fast it is to to run across all the data sets.
So how long it take a researcher to return the results?
I think it will probably take me, if I want to manually create this, maybe about an hour to 2 hours. And if I also try to consider the parental genotype, it would depend on how well the parental genotypes match with the progenies. If they don't match well, then it would take me a longer time to figure it out. I mean, separating the two problems of whether it's a true F1 and whether they match to a parent would simplify the process, and I think it would be much simpler and much faster if we do it that way. So
Carlos, I have two questions then. This is Kate. So I guess the first one is, so this, you are assuming that this would be the first step, but then you would do a second step where after you've identified the populations that you think are true F1s, you would then compare them to the expected parents to see if they're the desired F1s?
yes, you're correct. Kate, I actually forgot to mention that we can use actually this expected parent genotype with the actual parents and see whether they match them.
Okay, and then my other question was, in your algorithm, how are you splitting the populations? Are you using the sample group metadata?
Actually, the algorithm doesn't do the splitting. It's just another R script that does that for me now. But it should be taken care of by Gobi even before we put it into Flapjack. So even if we just try to import the Flapjack project file from Gobi into Flapjack, then once we run the algorithm, it shouldn't be a problem to use it from there.
Thank you, but it's using that particular metadata, the sample group.
Yes, you're correct. I think it's somewhere here.
Nice. Yeah, DNA sample group.
Yeah. I'm using that field too.
Thank you.
Yeah.
So in case the sample group or the crosses marked incorrectly, would your algorithm identify that? Like say you have 8 pollinations for each of the 140 crosses. In case mistakenly the crosses not the metadata entered, because you're trying to infer the parental lines based on the progenies within each cross. But if the progenies are assigned to the wrong cross to begin with, you won't be able to, from your analysis results, you won't be able to separate the individual assigned to the wrong cross. This is correct.
So is it just a subset of the sample star that belongs to another cross, or is it the whole?
Right. Occasionally.
So just a subset of the samples.
Right.
Yeah, that's correct. Actually, one of the one of the results here in the actual data, where there are 13 lines that were identified by the researcher and not by the algorithm, is basically due to that. So these are the 13 lines that were identified as a selfing by the algorithm, but was selected. No, sorry, I'm looking at the wrong filtering. So basically, we're looking for the 13 lines that was selected by the researcher and was not selected by the algorithm. So they only belong to 3 families, and one of them is this number 16 cross, and which is 3 individuals, 3, 4, and 8. And the other lines were identified by the algorithm as true F1 and vice versa for the researchers. So when we actually checked the population contained 2 different kinds of genotypes. So the algorithm identified these particular samples to be, these ones to be true F1, samples 1, 2, 5, 6, 7, 8, if I got that correctly. Okay. And the researcher identified actually these 3 of them, wherein we can clearly see there's a good relation between these three samples and within the other samples. So I think this is the case where in maybe these three or four are coming in from different crosses. The algorithm would just work which one is the most popular, and I think it considered because there are only three samples of this particular genotype, it considered the more popular. I mean the higher number of samples in the end. So was that does that cover what you asked, Star?
Yeah, this is very nice. Yeah, the supplement. Yeah.
Yeah.
So I consider you as very experienced doing this even manually, right? How about for the molecular biologists who are not very familiar with this process? Like say you manually may take you 2 hours, but for average people to do the curation, how long that could be? Or for the people on the line, like say Anna and Kate, Mike, you are very experienced doing pedigree verification, right? So manually, how long would that take you if you have 1,300 samples belong to 144 different crosses?
I have only done testing, this is Kate, with small numbers of things, so I've never tried to do such a large number of them, so I don't think I can give a time estimate, I'm sorry, but what he just showed in R is certainly faster than what I'd be able to do if I were doing this manually.
Hey, Star.
Hi, Mike.
You know, for us, you know, the major concern in Maze at least is checking to make sure the parents were correct rather than whether or not we have an F1. But I mean, I'm just trying to think. I mean, I've done it manually. You know, nothing. I mean, we're trying to move this to scale this up. In fact, we're just planning now to submit a bunch of F1s in the next month to check them. And I mean, you know, what I typically have done in the past in Excel, it'd take probably, I don't know, I mean, once I get the file set up, I've never worked on 1,300 records at a time. That would be pretty intractable. But, you know, a couple dozen at a time still take me an afternoon probably to, you know what I mean?
Right.
So it's, yeah, it's a slow process. And I mean, once I get a file set up the way I want to, but that setup takes time. And then, you know, the whole process is a bit tedious. So.
Thank you, Mike. I think the main thing is to put this in the broader context. We want to see the value of Carlos' scripts. Yeah, so from what he presented, I see this as the first step very quickly to find some mismatches or in order to dive into the details for the ones potentially there's some issue for off types completely derived from non-target parental lines at all, right? Then dive into the individual crosses. And then you will start using Flapjack, the existing F1 module, to look at more specific crosses.
Yeah, I think with Mike said, most of those functionalities of trying to see similarities between different lines is already implemented. So what we just need to do is take the output of this file. So both the selected F1s and the expected parental genotypes, and compare them with the actual parental genotype. So I think we're getting to that step, and actually that's a much straightforward analysis than this part.
And then a lot of times some parental lines not really included in the genotyping. You have to do the analysis anyway, right?
Mm-hmm. That's true.
So I guess, I mean, I was just mentioning, I mean, we should have a batch coming through in the next month. That would be somewhere around 100 markers with probably, you know, between 30 and 50 F1s that we want to check. I don't know if, you know, if it's something we could test.
Yeah, sure, Mike. I'll be willing to share the script with you, and I can help you run it. I can run it for you. So whatever you would prefer, and probably I could also implement the comparison to the observed parental genotype, and end up with a percentage similarity to the parent.
Mike, can I just ask, are these F1s between inbred lines, or are you checking test crosses with hybrids and inbred lines?
No, these are just source populations. F1s for that. They're populations we're planning to put through DH, but we want to confirm that the parents are correct before we do that. So
Great.
So I would just also like to mention that the another reason why I didn't want to include the similarity to parents yet is like for this example, for this particular data set, we ran into another pre-requirement that we need to end up with a consensus genotype. So I think for that we need to run another function to get the consensus genotype. And in cases that we do have like a reference genotype somewhere in the database, there's a lot of ways to get the reference genotypes or the parental genotypes, and that we haven't really figured out yet. And I think we're getting there step by step, and I think that's the next step after this one.
Okay, so I think we're coming to the end of this presentation, and thank you everyone for your questions, and please feel free to contact Carlos about his work in this area.
For giving your time to listen. Bye.
Bye now, thanks.
Thanks. Take care.